VC Chemistry, Unit 4, Structures of Protein. Okay, so previously we talked about primary structure. They are the number and type and sequence of amino acid units in the protein. It's the most zoomed in version of the structure. Okay, so we can actually see individual amino acids. Right. So it always starts with the N terminal on the left and finishes with the C terminal on the right. The entire shape of protein is first determined by the precise order in which its amino acids are joined together, because they all have their side chains, right? The way that the side chains stick out determines, firstly, basically determines in primary structure the shape of a protein that it's going to take in the future. Okay? So the function of a protein is a consequence of its shape. Therefore, the order of amino acid is extremely important in determining the function of a protein. Now, let me give you a math question. So since proteins differ from one another in number, type, and sequence of their constituent amino acids, right? So peptides with 50 plus amino acids are proteins. Now, so theoretically, how many different smallest proteins, so how many different peptides with 50 amino acids can there be in your body? Wow, jokes, you don't have to calculate that, but if you can, it's good. Well, but you can actually calculate this. If there are 3 amino acids, glycine, threonine, and valine are reacted to produce a tripeptide. So how many different tripeptides can they actually make? 3, 2, 1, ready? Yeah, it's 6, right? Because you rearrange 3 molecules in all kinds of ways, then it's 3 factorial, therefore it's 6. Well, what you can do is try to write them out, see if you are able to. Okay? Now, then we zoom out a bit to have a look at the secondary structure of protein. The secondary structure often involves coiling and pleating of sections of protein molecule that produces the secondary level. So that is mostly due to the hydrogen bond, only hydrogen bond forming between the hydrogen on the NH group and the oxygen in the carbonyl group. Okay. As a result, parts of the molecule might coil into a alpha helix, and beta pleated sheet. Now, alpha helix, it's the hydrogen bonding between the NH and the Cu on the same chain. This has to be very specific. If you are going to form a alpha helix, it has to be the hydrogen bonding on the same chain. Otherwise, it cannot really coil into each other, right? The reason that it coils is because the hydrogen bonding that brings them together. So this is like if you see a tertiary structure of protein, you can actually clearly see the secondary alpha helix there. Okay. Or sometimes there can just be hydrogen bonding between peptide links, not necessarily on the same chain though. Sometimes it can be different chains, but in this case, it can still be on the same chain. You just happen in a parallel fashion, not rather in a coil fashion. So this picture shows a more clear picture of it. So you can see the beta pleated sheet. It's because of the peptide links forming that hydrogen bond between CO and NH with each other at the same time. So you basically feels like it's 2 lines bonded together. That if you have like different lines all bonded together in that particular way, then it becomes a beta pleated sheet, somewhat like this. Okay. So then we zoom out even more, we will have a tertiary structure. So secondary structure is just alpha helix and beta pleated sheet. Then once you form the sheet and the helix, then this protein can still curl even more into each other, form a particular shape. And then that shape, that overall 3D shape, is called its tertiary structure. Okay, it's basically produced by the 3D folding of its secondary structure. Okay, and how do they actually fold together? This is important for chemistry. They are attractions literally of every kind that can bond things together. It's a lot of time is because of the side chains of each proteins can form hydrogen bonding, dipole-dipole attraction, covalent bonding when there is sulfur involved, then ionic interactions, and dispersion forces for nonpolar groups. So basically all kinds of chemical attractions we have ever learned can happen between the side chain of each amino acid interacting with each other. Sometimes it can also happen between the side chain and the main chain as well. So it's very diverse. So to form a tertiary structure, all forces are in the play. It's the 3D folding of the secondary structure. Okay. So in solutions, if the protein actually dissolves in solution, the hydrophobic side chains tend to fold inwards and so as to be away from water molecules. Okay. For some proteins, they look even more complicated. So once you already have a tertiary structure, you're already a proper protein, right? They can also, different tertiary structures can furthermore to stack on top of each other to form what we call a quaternary structure of a protein. Those proteins are normally very big. They're composed of 2 or more polypeptides or even interact with non-protein molecules to make larger and more complex units. Okay. So primary, secondary, tertiary, and quaternary. Quaternary structure does not happen to every protein. Only those big ones will have a quaternary structure. Most proteins will stop at like tertiary structure.